Okay, good evening and good morning, everybody. A very warm welcome to Soil Yourself September. This is a Flinders and Deakin University collaboration born out of our Grounded Minds Consortium, which we set up earlier in 2025 to explore and research the soil, gut, and mind connection. First, I'd like to acknowledge that we're hosting this series from Adelaide, also known as Tondanya, which is the land of the Ghana people. And I'd like to pay my respects to their elders, past, present, and emerging, and share my appreciation for their deep relationship with the land, air, and sea. This series is about something incredibly important, and it centers around soils. At first glance, soils are often seen as just the material that we walk on or in which plants grow. But dig a little deeper, literally, and it becomes clear that soils are the foundations of terrestrial life. They're inherently relational, so there's lots of relationships going on in soils, and they're conglomerates of physical, chemical, and biological components. And in terms of life, there's so much going on in soils, or at least there should be in healthy soils. So 59 to 99% of our planet's species live in these soil ecosystems. They provide our food. For instance, 95% of our calories are supported by soils. They filter our water and support biodiversity that makes ecosystems thrive. And the story doesn't stop in the ground. So the health of our soils is intimately connected to our own health. Now, from a big picture perspective, this is clear. As mentioned, we depend on soils for our food that keeps us healthy and alive. And there are many indirect roles that soils play in our lives. Now, a growing body of evidence shows us that what we eat shapes our health and well-being, both physical and mental. And we're constantly learning new things about this relationship. For example, you can refer to the field of nutritional psychiatry for how food influences our mood. And this field was pioneered by Professor Felice Jacka, who also co-founded the Grounded Minds Consortium. But there's also a growing body of science that shows us that microbes living in soils influence our health, influence the health of plants that we eat, and the microbes living inside us. These invisible friends shape our immune systems, regulate inflammation, digest foods, and provide us with nutrients, influence how our body's cells communicate with each other, and probably affect how we think and feel as well, via what's called the gut-brain axis. So this is a bidirectional communication system linking our gastrointestinal system to our central nervous system. In other words, healthy soils with all their diverse microbial life forms grow healthy plants, but they also help grow healthy people. Over the next month, you'll hear from a lineup of incredible speakers who we have scientists, practitioners, and thought leaders, each exploring different aspects of this soil-gut-brain connection. Now, some of the talks will be focused on the relationship as a whole, and others will be more focused on a particular component of the relationship, like one of last week's talks, which was on the gut microbiome. So there are 12 talks over the course of September, and it'd be great if you could attend several, if not all of them, if you're able to. And this way you can start to see the different components of this relationship and how they connect together. The talks will be recorded so you can watch them back on YouTube, and we hope that Soil Yourself September sparks conversation and inspires action. So thank you all for joining us on this journey, and I invite you to listen, to question and connect, and most of all, to think about how soils, guts, and mental health are all connected. And this is a big shout out to Ruben, who's also on the call today, who's been my partner in crime helping to organise this series. So thanks, Ruben. Now, everybody's microphones have been muted, but please add questions to the chat and we'll read them out at the end to the speaker. And please talk about the event on social media using the hashtag #SoilYourselfSeptember. And with that, let's welcome our speaker of today. He's the only speaker of today, actually. It's Dr. Craig Liddicoat. So Craig researches use DNA-based survey in soils and environments to help better understand the beneficial connections between microbes, soils, plants, ecosystems, and human health. In particular, he's investigating the restoration of health-promoting soil biodiversity for the People's City and Nature Research Programme in Aotearoa, New Zealand. More broadly, he works within the College of Science and Engineering in the Frontiers of Restoration Ecology Lab with myself, both collaborating on and researching activities involving restoration ecology, genomics, and healthy environmental microbiomes. So without further ado, over to you, Craig.
Thanks, Jake. You can hear me okay?
Yep, can you hear?
I'm going to minimize that little guy there. Yeah. Right. Very good. Yeah. So hello, everyone, and thank you for joining us. A big thank you to Jake and Ruben for pulling this webinar series together. My talk is titled Soil and Gut Microbiome Connections from Healthy Ecosystems to Healthy People. This little thing is annoying me. I'm going to try and move that away. Okay. I'll be focusing on how soil microbial communities shift with ecosystem condition, and also how they contribute to human health. In particular, how microbiomes provide a tangible connection that links the health of environments and soils to the health of humans. I'd like to acknowledge that my research is largely funded through the New Zealand-based People, Cities and Nature program, where our project team based across the University of Waikato and Flinders University is studying the restoration of health-promoting soil biodiversity. I just want to give you a bit of a roadmap for our journey tonight. The key route from healthy soils to healthy humans will involve supplying the right balance of macro and micronutrients into quality foods, and supporting, or in the unhealthy scenario, depleting the members of the gut microbiome community. And then there's many pathways between gut health and human health. So I will touch on this major route, if you like, but dare I say, we're going to be taking some dirt roads, getting off the beaten track. Sorry about the dad jokes. To look at how soils may be seeding, shaping and resupplying the gut microbiome, and also how they influence one health, affecting plants, animals, and pathogens in the environment, which also feeds back to us. So what is a microbiome? It's a community of microorganisms living in a particular habitat. Some words you might hear tonight. Microbiota refers to that community. Microbiome also includes the genes or DNA, the metabolites or breakdown products. It can also mean, also include their microenvironment. And metagenome, I'll pull that word out later, and that refers to the collective DNA. I guess over recent decades, we've come to learn that humans are not really a standalone organism like we think we are. We might think we are. We're really a system that includes microbes, and those microbes are a major part of us. Almost all of those microbes ultimately come from the environment, and especially strong influences in shaping our microbiome are how we're born, whether or not we're breastfed, our diet, and actually the surrounding environment, and we'll get to soils soon. And these microbial communities are very important for the normal, healthy operation of our immune system and for our gut function and metabolism. So how do we really know that gut microbiomes have such a huge influence on our health? Because studies in animals and humans show that transplanting gut microbiota can transfer disease or offer protection from disease. A recent study compiled a list of conditions for which registered clinical trials were underway. This is as at 2023, bearing in mind there would have been already some level of evidence before moving to human trials, but medical researchers are looking at microbiome transplants to treat inflammatory bowel diseases, irritable bowel syndrome, obesity, diabetes, cancer, fatty liver disease, multi-drug resistance, depression, neurological disorders, among other diseases. So some key roles for gut microbiota are that they have a functional repertoire or can produce a vast range of metabolites that's well beyond the capacity of human digestive enzymes. So the microbiome is like the middleman. If we want to extract all the good nutrition out of our food, that is largely down to the microbiome. So our diet combined with microbiome and metabolites stimulate hormone secreting cells that line our digestive tract, and they trigger signals and processes that regulate appetite, growth hormones, release of bile acid, metabolism of fats, and gastric movement. So what are things to look for in a healthy microbiome in our gut and perhaps also in soils? Bearing in mind the science here is not totally settled, but here are some likely candidates to look for. There could be key taxa or key organisms with known beneficial roles. In soils, think of nitrogen-fixing bacteria or mycorrhizal fungi, things you can look for and notice when they're missing. There might be indicator groups that are associated with healthy or disease states, molecular patterns. Bits of microbes can trigger immune responses. Particular constituents, the sort of molecular or chemical compounds that are released when microbes die, may also be taken up and used in health promoting pathways. And then there's the functional capacity of communities, or their potential to metabolize various different types of compounds. And this might occur through a string of activity or collectively in extracellular space, so outside of the bodies of microbes, where they have shared resources to perform certain roles. So these communities can have multiple tens of thousands of species and multiple tens of thousands of functions that work across those species. So that type of complexity suggests that community scale effects are going to be important. If we move now to soils, if you hadn't heard, soils are home to an estimated 60 to 99.9% of species on Earth, making them the richest environmental microbial reservoirs. And operating across living root zones, decaying residues, and bulk soil, microbial communities, they form and they decompose, they recycle soil organic matter, which becomes a primary source of the macro and micronutrients that we hope make it into our food. So collectively, microbes and organic matter are important in really to growing nutritious food. Microbial communities will shift as plants grow. Root exudates or sugars attract and recruit certain members from the bulk soil. This subset of a larger pool of species is favoured. It builds microbial biomass and it establishes particular processes and networks of activity. With increasing above ground diversity, there are more complex interactions below ground, more interactions between different types of root zones, and greater diversity of habitats. Some of these different plant species will work better together. For example, in the diagram here, the grass helps to stimulate the rhizobial nodulation, helping the legume to fix nitrogen from the atmosphere, some of which makes it back to the grass. So each gets a benefit from helping the other. And many microbes can only survive in communities because they can't do everything they need on their own. Microbes and nutrients can have very important interactions. If we look at just the key nutrient of nitrogen, for example, we know that inorganic nitrates and ammonium are used by plants. However, we've come to learn that plants can also use more complex, chunkier organic forms of nitrogen, ranging small to large, from amino acids, peptides, proteins, and even whole bacteria. So these are kind of like ranging from organisms to bits of organisms that break down. And when those chunkier organic forms are being used as nutrient sources, perhaps there's also likely to be other macro and micronutrients coming into the plants. When whole bacteria are consumed as a source of nutrient, we call this rhizophagy. And interestingly here, excessive fertilizer regimes can have a detrimental effect on soil microbiomes. With excess nutrient, plants are losing the incentive to invest in root production. They're losing the incentive to invest in those fungi, root, or mycorrhizal associations. So, there may be opportunities here for farmers to save money by dropping back excessive fertilizer inputs and letting the microbiome do its thing. A word of caution, and no need to tell farmers this, but not all soils are the same. Soils can be naturally different. We see here some soil profile images here of very different soil types. Not all soils are the same. So yeah, soils can be naturally different, and also they can vary, in terms of poor health versus good health relative to their natural baseline state. It's worth noting that physical and chemical conditions can either support or put a handbrake on microbial communities in terms of the habitat, activity, and diversity that they can express. For example, high organic matter and clay content in the soil on the left provides premium microbial habitat, whereas acid or alkaline soils, salinity, and infertile sands can represent barriers to building diverse soil microbiomes. Just briefly to note the main methods that our research group is using. These are community scale approaches. From soils, we can extract DNA, then use either marker gene or amplicon methods, which target a particular gene region. I'm going to have a drink of water here. targeting particular gene regions useful for identifying taxonomic variation. Some may have heard of bacterial 16S rRNA or fungal ITS gene regions. And from there, we can characterize and interrogate who is there and get some reasonable information out of that. The other approach, which is a bit more powerful, involves randomly chopping up the DNA from the whole community. And the DNA provides information on functional genes where we can examine not only who is there, but what can they do or their functional potential. So we've studied changes in soil microbial communities, community composition in degraded ecosystems as they're restored towards natural reference states. And the example here is from a post-mining forest restoration in Western Australia. The y-axis is a measure of how similar the community is to a natural reference. And on the x-axis is the age in years since restoration planting. In these forest ecosystems, it can take several decades to return to natural levels of variability, just in terms of the soil bacterial communities. Looking at crops and pastures under biological focus versus standard district practices versus native vegetation from some South Australian farms. This is from my own unpublished pilot scale study data. We saw overlaps and differences in communities across bacteria, fungi, protists, and invertebrates. The two-dimensional plots here are a way to visualize community scale differences. Every point is a sample. The closer they are, the more similar, and points that are far away from each other have very different community compositions. The colors here represent different groups. So orange, standard cropping, pink is standard pasture, blue, biological cropping, purple, biological pasture, and green is native veg. So what I've hopefully conveyed so far is that gut microbiomes are important to our health. Also that soil microbial communities support nutrition getting into our food. And those communities can shift with land use and management practices. Now I want to spend some time further linking soil health and human health. And broadly, this fits into the area of One Health. That's the interconnected health of soils, ecosystems, plants, animals, and humans. And you might appreciate this is a very broad area. I won't attempt to cover all of this, but just to sort of set the scene, I'll draw your attention to this little caption here under the left-hand panel, which says microorganisms as the conduit of One Health components and soil microbiomes as the microbial reservoir. And in particular, we're going to take a closer look at connections between soils and humans through the microbiome pathway. So over the last 20 to 30 years or so, a growing number of medical researchers and immunologists have been advancing the biodiversity hypothesis. This is the idea that loss of exposure to natural microbial diversity through biodiversity loss, soil and land degradation, and urbanization are contributing to a rapid global rise in allergic, autoimmune, and chronic inflammatory diseases. Becoming deprived of microbial exposures, our immune systems have become poorly trained, unfit, and dysfunctional, so the theory goes. Especially in urban areas, many have largely lost contact with soils, and soils from urban green spaces have become more homogenized globally due to similar green space types and management. They've become home to more fast-growing, opportunistic, potential human pathogens, more antimicrobial resistance genes, and communities that are better adapted to faster nutrient cycling and more stress. Other key phenomena happening in parallel are that modern industrialized diets, high in fat and sugar and low in dietary fibers, are causing the extinction of important members of gut communities. And this can get worse over generations. It looks like I'm picking on hamburgers here. I guess you can make healthy hamburgers. Meanwhile, science also shows that a major share of our gut microbiome comprises spore-forming bacteria, and these can activate in the anaerobic gut environment, but also protect themselves when they're outside in the outdoor aerobic environment, where they can redistribute, potentially also finding a sometimes home in organic, rich, and sometimes wet soils. So hang on, spore-forming bacteria, that kind of sounds bad. And aren't bacteria bad anyway? Like in hospitals and with immune compromised patients, that's the right attitude. Looking at this conceptual dose health response curve, it's not really a curve, it's a straight line, but with a dose on the of some microbe on the x-axis and health response on the y-axis, we kind of think linearly in that excessive exposure or dose will probably lead to infection and disease. And in hospitals, generally speaking, we can probably agree that the only good microbe is a dead one. But maybe this is not the best template of linear thinking to take into everyday life. Not enough. Compare this sort of thinking with something a bit more nuanced, that not enough exposure to a naturally diverse range of microbes is leading to a poor immune training and immune dysfunction. So we get negative health outcomes. Somewhere in the middle, where we have the right kind of exposures to certain microbes with levels of biodiversity that keep pathogens in check. And I see the analogy here to this generalizable U-shaped optimization curve that we see across biology, especially in plant nutrition, this idea of going from deficiency, sufficiency to toxicity. So it's really tempting to ask, are we too clean? And the image here is a bit of an extreme example. It comes from a conversation article describing microbiomes of hunter-gatherer populations. The person has just prepared an animal for consumption. They're cleaning their hands with the microbe-rich stomach contents of the same animal. So that's pretty gross, sounds pretty gross, and is far from our modern ideas of hygiene. But, and I'm not recommending this, but let's think about the fact that humans have co-evolved with microbes, all sorts of microbes. We're potentially building immune fitness here, and this person is using the microbial diversity to counteract potential pathogens. So microbes coming from the environment, coming from soils, can find their way into the gut and help to boost our health through some key pathways. They can act in supplementing our protective microbial communities, not leaving room at the table, if you like. They can participate in immune signaling, triggering defensive inflammation, also triggering tolerance. They can help build immune memory, and they can provide these beneficial functions or metabolites. So we have this idea of two layers of protective biodiversity. If we're a child just starting out in life, or older and our microbiome is perhaps depleted through poor health, the outer layer of biodiversity in the environment and in the soils, soils are a key part, provides an important natural reservoir for supplying and resupplying microbial diversity to our human microbiome. In fact, animal studies show that soil exposure modifies the gut microbiota and builds immune tolerance. Intervention studies with daycare children, where researchers brought soil and plant materials into daycare yards, found changes to their microbiomes and improved biomarkers for more tolerant immune systems within 30 days. Just on the left-hand side here, there's two studies. On the left-hand side, studies of asthma and allergic risk in kids growing up on farms versus cities showed lower rates of asthma and allergic risk in the farm-exposed kids. In the right panel, comparing traditional Amish farming communities who would have had greater exposures to soils and animal manures versus Hutterites who employed more industrialized farming practices. They showed lower rates of asthma and allergies in the Amish farm kids. In mouse model studies, soil exposures help to accelerate gut microbiota recovery following antibiotic treatments. And as I said, soils aren't all the same, so be careful with what soils you're being exposed to. In another mouse study, farmyard type housing materials provided a level of protection against the onset of colorectal cancer. In separate studies, researchers have found that soil biodiversity helps to suppress pathogens and reduce the persistence of antimicrobial resistance genes. Further to this idea that not all soils are the same, this slide is from one of my PhD chapters where I examined a proxy measure of soil microbial diversity. That proxy was actually a thing called soil cation exchange capacity, and that reflects organic matter and clay content in soils. And I found that in the lower socioeconomic groups, we'd expect to see environmental influences on health stand out the most, that hospitalisation rates for infectious and parasitic diseases, which are rising on the y-axis in this plot, they were the highest in regional local government areas with typically low microbial diversity soils. So these results suggest that areas with high microbial diversity soils may help boost the immune fitness of populations living there. Anxiety-reducing responses have also been found from exposures to the soil bacteria Mycobacterium vaccae, and this study has previously gained a lot of exposure in promoting mental health benefits for gardeners. In my own recent work, still under review, so not yet finalised, I've been exploring the question whether exposure to degraded ecosystem and soils, which are expanding globally at alarming rates, may be linked to metabolic alterations that are found in type 2 diabetes, because type 2 diabetes is really a metabolic disease. And you may be aware that the origin of type 2 diabetes is not totally understood, and it's also rising at alarming rates. There are studies where the symptoms of type 2 diabetes have been corrected using fecal microbiome transplants. And in simple terms, this is a disease involving altered energy harvesting and altered energy management. So I've been working on a method that characterizes the functional capacity of soil and gut metagenomes, then redistributes that genomic potential to estimate a theoretical capacity of those communities to metabolize or to work on an array of chemical compounds. And the reason I'm interested in translating it into chemical compounds is that historically we've gathered a lot more knowledge of health associations with the metabolism of chemical compounds compared to tens of thousands of poorly characterized organisms. And these compounds are like the currencies or the key resources in the microbial economy. So we might gain insight into what potentially valuable resources are being exchanged and processed across the communities. The data here show potential metabolism of compounds on the y-axis across three data sets. The first data set showing the lines is a soil metagenome data set from post-mining ecosystem restoration. We've got restoration age on the x-axis. And then the other two data sets are gut metagenome study data sets in type 2 diabetes and versus normal healthy controls, and they're from Sweden and China respectively. So from thousands of compounds that could be being worked on by these communities, these three groups of compounds stood out with common trends across the soil and gut data sets. In the most degraded soils, which are indicated by the blue and red arrows there, in the most degraded soils and in type 2 diabetes, they shared elevated potential metabolism for certain sugars and decreased potential metabolism for lignin or plant-based fibers, and also decreased potential metabolism for an interesting group of molecules called branch-chain fatty acids. So these exploratory findings remain to be tested further, but if this is a thing, what might be going on here? If we compare an intact, more natural ecosystem on the left versus a quite degraded ecosystem on the right, generally speaking, there'll be larger reservoirs of soil organic matter in the more intact systems, more plant debris, including lignin-type fibers, and more recycling mechanisms. Whereas in the degraded system on the right, there's typically a loss of soil organic matter, which ultimately breaks down into simpler and simpler sugars, especially through the action of opportunistic microbes that will enjoy those conditions. And this could explain the increased potential metabolism of sugars and decreased potential metabolism of lignin, which may be transferring an altered metabolic profile to the human gut if people are experiencing greater exposure to degraded ecosystems and the microbiomes that spread from them through the aerobiome, I guess. And the monomethyl branch chain fatty acids are interesting. I hadn't really heard of them until the signal popped up in the results. However, they are known in nutritional science and they're known by researchers of metabolic diseases. The signal here was actually for synthesis of these molecules, which occurs in bacteria, as these monomethyl branch chain fatty acids are a key component of bacterial cell membranes. And they're precious molecules. They're recycled and transferred across bacteria, fungal, and animal domains. They're synthesized from branched chain amino acids. And interestingly, those branched chain amino acids are released from soil organic matter. And the branched chain fatty acids are also recycled and made longer and longer. And they can be recovered and recycled through, for example, fungal recycling networks. As I said, they're quite precious, and they're used across bacteria, fungi, and animals. So these branched chain fatty acids might be incorporated at high or low concentrations into bacterial cell membranes, and they may be released into the gut due to normal rates of gut bacteria turnover. In the gut, these fatty acids are taken up by human host cells, where they're involved in lipid metabolism and also the central regulation of human host cells. They're in fact needed to trigger a particular mechanism called the mechanistic target of rapamycin complex 1, mTORC1. So that's a mouthful and it sounds super complicated, but what the important thing is that that's a mechanism that when it's not working properly, it's associated with diseases including diabetes, cancer, and neurodegenerative disorders. So I guess stay tuned what happens to this study. It is exploratory. However, there's some interesting potential mechanisms that could be operating there. So sort of nearly wrapping up now, I came across this recent paper describing therapeutic approaches to boosting gut microbiome health. And I couldn't see soil in here, so I thought I'd provide a bit of an overlay. We've got prebiotics, so they're kind of like with living organisms. The probiotics are what the organisms use, the probiotics are the living organisms. The prebiotics are what they use. Symbiotics are a bit of combination of those. Whole diet, fermented foods, you can probably work that out. So here are some dietary components that will rely on healthy soils. Here are some further components that might be cost effectively supplemented through ambient, perhaps occasional light dust exposure to healthy soils. So yeah, soils are potentially part of this story and might be used for cost-effective health benefits. So in conclusion, I guess regenerative practices that focus on building and protecting soil organic matter and soil biodiversity will shift microbial communities and their functional capacities. Microbiomes are key to supplying macro and micronutrients and hopefully getting that into our food. Biodiversity and reduced disturbance are associated with greater health protection and progressing towards One Health approaches. Soil and gut microbiomes do have a degree of overlap, not obviously not entirely, but there is a degree of overlap there. And exposure to naturally diverse microbiomes from soil plant systems show health benefits through immune training, seeding, shaping, and resupplying gut microbiome and functions. So that's it. I know I've gone through a lot there. Hopefully I've promoted a greater appreciation of the connections between soil and gut microbiomes and healthy soil ecosystems to healthy people. So what if I stop sharing there, Jake?
Yeah, you can do. Thank you very much, Craig. That was fantastic as always. I forgot to mention that Craig's also a key member of our Grounded Minds Consortium as well. So thanks, Craig. So let's look at some questions in the chat. So one question is, how do microbes on above ground parts of plants tie in with One Health?
Yes, I mean, I guess you'd be looking for diversity there or perhaps microbes with key beneficial roles. But when they're on the above ground parts of plants, potentially they're making their way through grazing animals. They might come through in raw foods. If they're working their way inside to the interior of plants, then potentially they're coming through the food as well. But I think what we're seeing as well is that microbes can move their way from the soils, be taken up by plants potentially, or blow up on plants, they can then drop in the plant debris, in the leaf litter, and then be part of that community there on the surface and in the upper soil layers. So there's definitely pathways that microbes on plant surfaces can make their way into that broader system of through our diet, through grazing animals, the health of the whole system. Yep.
Thanks, Craig. This sounds like a bigger problem that I can address in my backyard. What should we be encouraging our politicians to do to improve soil health?
Yeah, so obviously, that personal exposure to healthy soils is part of your own sort of personal health benefit. So, if you've got a backyard, I'd encourage doing something. I'm probably should do more myself in that regard. But yeah, sort of onto the what do we do in terms of bigger picture, the food system as a whole. It's definitely I think a work in progress, and there are groups. There's a regenerative food systems conference in Perth next week, looking at this exact sort of topic. How do we go from soil health, food systems, to really connect up those ideas better than we are at the moment. And farmers might get, I'm not a farmer, I can't really sort of comment on farming too much, but if there's opportunities for farmers to save money, cutting back on over fertilizing and helping the soil microbiome do their thing, that could be a win-win, really. And I hope there's opportunities to, it's a work in progress essentially, so more needs to be done.
Thanks, Craig. I think this Grounded Minds Consortium probably plays an important role as well. So actually demonstrating to politicians the importance of that connection, hopefully will help them to do something. So what's the next question is, hi, what are the best models to study microbiome studies?
The best models?
Did you want to just perhaps if you could clarify that question in the chat, and then I'll re-ask that again in a minute, just provide a few more details for Craig. I'll just move on to the next question in the meantime. How are soil contaminants like microplastics affecting the soil microbiome and in turn our own microbiomes?
Yeah, so obviously I didn't go there. I didn't go at all really into contamination and microplastics, but I guess you could sort of put them into that the concepts of how they impact on physical and chemical conditions in the soil, and potentially they're providing a barrier to I guess allowing a more diverse community to develop, and perhaps degrading the habitat that's available there. So yeah, they can potentially put a handbrake on what can happen in the soil microbiome.
Yeah, good point. Good question as well. So often we're saying, all the kids should be playing in the mud and it's really good and we're too clean, etc. But then we always have to caveat that now with, we've polluted the environment so much. So there's things like heavy metals and microplastics, etc. in the soils. So I think it'd be really beneficial for someone to develop some kind of easy to use soil test for the general public to find out if the soil might be, really toxic or not. But
yeah, and the other, I guess, as an extension there, for activities like bioremediation, I guess we might be encouraging certain microbes that do well in those contaminated soils or with plastics or whatever to get in there and do a job to break them down into more friendly sort of forms. So yeah.
Well, thank you so much. Another question from Jane. Great talk, thanks. Great. Regarding microbial exposure via soil and food, are the microbes in our food, are they actually seeding our gut microbiome, or are they usually transient? Yeah. Can you increase your gut microbiome diversity as an adult through diet?
Yeah, great question. I mean, this concept of the transients, and that's definitely a thing. You can have microbes that just go through temporarily, and they can have an influence. They can trigger immune responses. They can push the community in certain directions, and then not necessarily hang around. But I think some of the members that you will find in there do make more of a home in there. But remember the comment, the study that I pointed out where they're talking about spore-forming bacteria. So you're going to have these spore-forming bacteria that like it in the anaerobic gut, but they're not necessarily going to hang around permanently. You'll sort of get a turnover where they come and go. So they're probably at home in the gut, but then designed more or less to not just sit in there permanently, but to come and go. So, and also the facultative anaerobes that can handle a bit of more higher oxygen levels, and then towards the really low oxygen levels. So it's a bit of a mix. You're going to have transient ones, and you're also going to potentially have ones that hang around a bit longer.
That's on Craig. It's probably important to point out as well that the gut microbiome is most plastic, so most easily colonized at the kind of weaning age between 0 and 3 years old. So the diet and other exposures are probably going to have a much higher impact during that critical window of development.
Yes. Yep.
Ruben, over to you.
Thank you so much. Another question from Ramu. What about the hydroponics? They never link to soil, so how much it's healthy in terms of not coming from soil?
I've actually wondered that myself. It's a great question. I don't have the answer, and potentially there's microbial communities in hydroponics systems anyway. So if yeah, it's a great question. I don't know. It depends on what sort of setup there is for the hydroponics as well. Like if it's sort of going through a bit of a wetland type filtration system, you might be getting some sort of environmental microbes in there that are used to aquatic conditions and maybe spend some time out. Great question. I don't have the answer, but I'd really like to know because, yeah, we see hydroponically grown tomatoes here in our sort of city of Adelaide here. I'd like to know. Great question.
Good question. Craig, do you know if these hydroponic systems, the environments are highly sterilized or not?
I can't even say that. Yeah, I mean, I guess they'd want to reduce sort of fouling. So maybe I don't know if they sterilize or not, but they'd want to reduce sort of fouling through their pipe works and all that sort of stuff. Jake, you mentioned before the sort of critical window when in infancy and that sort of thing. And I just wanted to sort of highlight something I read linking to that the study there where I found the monomethyl branch chain fatty acids. So they're really rich in bifidobacterium and really rich in breast milk. And the I forget the technical name, but the lining that the baby comes out in when they're born, it's really rich. These monomethyl branch chain fatty acids are rich in that. So I don't know, there's something really interesting there.
Interesting. Yeah, for sure. And just say that Ruben is actually leading a systematic review on the potential of colonization by environmental microbes in the gut. So if anyone's interested in that, watch this space probably next year.
Yep.
Ruben, did you want to ask the next question?
Yeah, the next question from Penelope. Hi, Craig. You mentioned there is a degree of overlap between soil and human gut microbiomes, but these could just be ubiquitous types of microbiota which naturally co-occur in both ecosystems. Has it been proven that these soil microbiomes actually survive human digestion, interact with the gut microbiome, or even colonize?
Yeah, so colonizing is I guess if we wanted to look into it in heaps of detail, how do you define that? Does it have to hang around forever? Does it sort of does it hang around for a certain length of time and then sort of go? So and some of these organisms, for example, in my PhD, we did a mouse exposure study where we exposed these lab mice to soil dust, and we had no soil. We had a high and a low biodiversity macro and micro soil that we exposed these mice to, and we looked at changes in the gut microbiome of these mice and what changed over the course of a 7-week exposure. And there was a whole bunch of just mouse intestinal bacteria and looked pretty boring. And there was one particular organism that stood out that was across all of the mice. but it was most supplemented in the high biodiversity soil exposure. And it was an organism that was first characterized in a soil plant system. And it was a spore-forming anaerobic butyrate-producing bacteria. So yeah, this was first described in a soil system, and yet here we found it in all of the mouse guts. So
was that the Kiniothix
yeah,
adasoides?
yeah, that's right. So to the question, there's probably not enough effort that has been thrown at really getting to that question of all of the sort of organisms that are found in the gut, how many of those could also have a home in soils? There's probably not been the really the money, the resources, the research effort thrown at that question. But you know, I'd put money on the fact that some of those things can have a home in the gut, and some of those things also, you know, have a home in soil or in the environment. Because when we lose all our, when we're going to lose our gut microbiota, which we do, where do they come from? Where are they going to come from to be resupplied? It's from the environment somewhere.
Cheers, Craig.
Thank you so much. Another question from Ariane that is also part of Soil Yourself September. So thank you so much, Ariane, for joining us today. Amazing tag and work. Thanks, and I'm interested in the impact of branch fatty acids on rapamycin regulation and immune system functions. Might you elaborate? And a technical question is it possible to estimate soil metabolic function in terms of rates from shotgun metagenomic studies?
Okay, this is getting quite technical. So the particular method that there's probably more widely recognized techniques to go from the shotgun metagenomics in a defined environment like the gut. So you would sort of assume certain environmental conditions, and then with, I think, the sort of organism-specific models that sort of model rates of the chemical compound metabolism. So there are probably more rigorous approaches in very defined scenarios, and that's not really what I did. I took the shotgun metagenomics. You can take that through to functional potential, and then I linked in reactions and chemical compounds to that to more or less do a hypothetical, what is the hypothetical distribution of that genomic potential across all of the chemical compounds that those genes are theoretically able to work on. And so that's kind of what I did. So it's really a different sort of a new theoretical way to look at the capacity. So, and I use that in soils because you can't really assume an environment in soils. Like in the gut, you can assume fairly limited conditions, you know, the low oxygen, the fairly well-defined chemistry, pH, etc. Whereas in soils, the conditions are so variable that the communities in soils, their capacity, the communities are responding to the available resources and the types of environments that are happening there in soils. So I really designed my method around the idea that soils are going to be quite variable. We'll have to chat offline, I think.
Yeah, good one. And metabolomics can be used, can't it, to get like a direct snapshot of function in the soil? But obviously,
that's right.
There are caveats, like you say, it's highly variable, high complexity, context dependence, etc.
Yep.
Great. Another question, very interesting from Judy. Soil has microbiomes. Does the seawater, as a large part of planet's surface, do they live there too? For example, food like seaweed contain it. Yeah, that's an interesting one.
Yeah. I think I mean, I've not studied it, but as far as I know, definitely there are microbiomes in the ocean. You know, and there's different habitats in the ocean, in sediments, on seaweeds, in and around algae, a whole bunch of different type of microbial life. It's obviously a lot saltier, so yeah, that's like things like pH and salinity. They're key I guess, selection filters on what type of microbes can survive and proliferate in those different types of environments. So, yeah, I mean, Jake, what do you think? My gut feel is that the communities would be quite different.
Totally. Well, the communities will be very different, but I think microbes in the ocean make up about 90% of the biomass of the ocean. So there's definitely lots and lots of microbes living in there. They'll be pretty different based on, yeah, exactly like the environmental filters that select for certain species and functions. Good question.
Yeah, well, another question from Jane. Is there evidence that a soil transplant from healthy biodiverse soil can speed up microbial recovery of depleted soils in conjunction replanting rehabilitation projects?
So this is the idea of like inoculating soils. If you've got a very if I'm hearing that correctly, that's like you've got a very degraded soil. How do you get a healthy soil microbiome into that system? And I guess this is kind of a bit of an area of ongoing research, different ways that you might try and inoculate or introduce microbes into that environment. You're going to have, and we're really interested in this, Jake and I and our team are really interested in this idea, how we might do this better, or some of them are going to be followers and some of them are facilitators, if you like, working in and around different plants that might establish, and you're going to have succession in those plants. Maybe there's a succession that's happening in the microbial communities. So, you might sort of spread a dusting on the surface. You might want to try and translocate more intact soil profiles as sort of islands that can be used to colonize further out. Bacteria and fungi are going to have different approaches, I guess. So it's an interesting question and something that our group is, really quite interested in researching and more.
Thanks, Craig. Any final questions before we wrap up? Rami has a interesting observation. He says, in some Indian traditions, the dried umbilical cord stump is preserved and used later in milk for healing, especially in cases of illness or to treat infertility. Thanks, Rami. Interesting.
Jake, there was the question before about the models. What type of models are better?
Yes.
So I'm just sort of thinking some thoughts, like mouse models where you can do a lot of controlling of all the variables are quite useful if you want to test a particular treatment or idea. So that's why mouse models or some animal models are used to test different microbiome treatments. But I guess they can be a little bit different when you're really interested in humans. And so when you're looking at human trials, you need to boost up the numbers to get more evidence of effects. So that's probably just something on the models that you might use to test microbiome.
Yeah, good point. And you can, if you search on YouTube for Alice Martin's talk in the Soil Yourself September, she did talk about organoids as well. So these are kind of tiny simplified versions of organs that are grown in the lab. And this has the benefit of being more ethical than using mice models and it prevents the kind of the cross-species complexity as well. So you can have human-made organoids to test on.
Yeah.
Okay, I think we should wrap up unless anyone's got anything else to ask. Getting lots of positive comments in the chat, and I agree with everybody. It's fantastic talk. Thanks, Craig. Thanks so much.
Thanks everyone for joining.